I want to turn the focus to sex chromosome evolution. As we all understand, and most the consensus view of the sex chromosome evolution is the sex chromosome will evolve from a pair of ordinary autosomes. But we are very interested in how this process happened, the detailed process of the evolution mechanism. And usually in the model, there's a sex determination factors. So the location of the sex determination factor triggered the differentiation of the two chromosomes. But just the sex determination gene itself is not sufficient. There are other features of this diverged sex chromosome. And one particular is lack of recombination between the X and Y chromosome or Z and W chromosome. And so the recombination or suppression of the recombination is one of the important steps during the evolution of the sex chromosomes. To understand this, I think the best example is the human X chromosome. The human X chromosome and Y chromosome are diverged for a very long time, as illustrated on this slide. I should put like the pair of the X and Y, but I want to explain this first. So on the X and Y chromosome in humans, they are a stretch between the X and Y. They are still recombining, and like even now. So we call it a pseudo-autosomal region. So compare the rest of the X and Y chromosome. In that small region of the XY chromosome, they share some homologous genes. So that's why we call it the pseudo-autosomal region. But largely, the genes are totally different between the X chromosome and the Y chromosome. Showing on this slide is to talk about the evolution of the chromosomes and based on the divergence between X and Y. So starting from the sex determination factor, that's the XRY, then the region around that, the chromosomal region around the sex determination was inverted. So the inversion suppressed the recombination between the proto X and proto Y. We'll see the mechanism a little bit later. Then the genes in these regions are diverged during evolution because they can accumulate different mutations independently. So I think this slide is more clear to talk about or to illustrate our hypothesis how the divergence took place between a pair of sex chromosomes. So in the beginning, there's a pair of autosomes. There's a sex determination. Showing here it is the XRY. Then in other lineages, like monotrins, we still can see the XY chromosome like illustrated in the middle of this slide. So by comparing different sex chromosomes in human and the other lineages, we were able to reconstruct the phylogenetics or phylogenetic history of the sex chromosome. Then the second one in blue and yellow is more similar to the XY system in marsupials. So showing in this slide, and the number of years, like how many years of divergence was on the top, just to illustrate how we think the evolution of the sex chromosome in mammals was evolved from a pair of autosomes. Then on the X chromosome, there are different regions named 1, 2, and 3. And in corresponding to the region on the Y chromosome in blue, that indicates the regions was the recombination of this region was suppressed. So showing in the first slide, you can see the first, like the region 1, which contain a large region, and at least in monotrames, this region compared to the Y chromosome are largely diverged. Then there's a second region we recognize in marsupial, and that also in the surrogate. The second stage, this small region was become diverged between the X and Y chromosome. Then the third and the fourth. And all this analysis will start with the Y chromosome in human, because we used to think Y is important because the Y chromosome, the males with normal Y chromosome develop normal fertility. But we didn't recognize how many genes exist on the Y chromosome. We think Y chromosome is highly reduced, and also there's not much genes on the Y chromosome until we look at the genomic sequence of the Y chromosome. There are genes discovered on the Y chromosome, and we can compare the divergence between an XY to reconstruct the evolutionary history of the sex chromosome evolution. So largely we recognize from this slide and also several other studies, the divergence of XY took place for a million years. So in most XY systems in different lineage, they are totally different. And in general, Y contain only a few genes compared to the X chromosome. But in some lineages, Y chromosome is important for sex determination, like in mammal, or it's important for male fertility, like in Drosophila. So the XO male in Drosophila are viable and has the male feature that's like a morphologically male, but it's infertile because the lack of the essential genes on the X chromosome, on the Y chromosome for male fertility. If we put the information together, I want to explain in this slide from different organisms. Okay. So the first one is the evolution of the human Y chromosome, the comparison between the X and Y. On the X-axis is the position of the chromosome, and this in scale to what we call the pseudo-autosomal region. That's showing in the part here. Like, so the red, or like the thick red or blue part. And I already talked about the first part of the human XY chromosome. Basically, the Y-axis is the divergence between the X and Y. And showing in this slide can show the divergence between the two chromosomes is smaller when the region is closer to the pseudo-autosomal region. So that also corresponds to our hypothesis, and that region was late, like the last part include in the divergence. So that means the lack of recombination between the two chromosomes is more recent compared to the rest of the chromosomes. So that's the different stratus. And this is showing on the first slide for human. And very similar phenomena were also observed in the chicken and also in plants, dioecious plants. So in chicken, there are ZW system, very similar to human, but the heterogametic sex is the female. That's what we talked in the beginning of this lecture. But if we look at what we think about the divergence between the ZW chromosome, and the pattern is more or less similar to what we observe in humans. That is, the regions close to the pseudo-autosomal region are less diverged than the region far from the pseudo-autosomal region. But you probably also notice there are regions with reduced divergence in this slide, which is away from the pseudo-autosomal region, the PAR region, which is because the inversion occurred on this chromosome. And in dioecious plants, plants, when we learn plants, we always look at the flower and the flower has both male and female organ. But in dioecious plants, they have sex chromosome. They have different male flower and female flowers. So the study of the sex chromosome in dioecious plants, they are also XY system. So they are X chromosome and Y chromosomes. And if we compare the divergence between their XY chromosome, and we also observe the similar phenomena as the divergence in the human XY chromosomes. So the evolutionary strata seems like a general observation in different lineage. Regardless, this sex chromosome all evolve independently because they are all very different and the sex determination are different as well. Okay. So now we want to talk about what happened during evolution. We already observed there's large degeneration in the Y chromosome in terms of number of genes and also the sequence of the Y chromosome. So this slide is from one of the review papers, try to talk about the evolution at the sequence level and how that evolved to the strata pattern. So in this slide, we can see they divided the evolution of the Y chromosome into several steps, and similar to the previous one, but the previous one was showing, okay, this was hypothesized in historical events, and we still can recognize this type of Y chromosome or XY system in other lineages. But here, the hypothesis was, okay, what happened in the sequence and molecular level between or during the evolution. So the first one is the proto X and Y. They start, so we skip the very first one. They start from an autosome, a pair of autosome, and there are sex determination factors located on this chromosome and which cause the beginning of the differentiation of these two autosomes. Now we call this proto X and Y. So the proto X and Y, the characteristic for the proto X and Y is the sex determination factors, and sometimes in combination with, for example, male sterility and female fertility. It means only the homozygous of this proto X chromosome can produce the female, but the proto X and Y together produce the male. And during evolution, the recombination suppression was initiated by inversion. So if the region is inverted between the two chromosomes, and that particular region will not be recombined, or the genes, the genetic material in this particular region will not recombine between the two sex chromosomes. And this will allow the accumulation of deleterious alleles, including transposable elements. The reason we hypothesize that is we often observe repetitive sequences on the Y chromosome, and also a lot of them are transposable, similar to the transposable elements. So now you can see from stage 2, 3, 4, the regions or the degeneration regions on the Y chromosome or on the proto-Y chromosome become larger and larger. Then accumulation of the deleterious alleles become more and more. And that results to the reduce in size for the Y chromosome. They try to show that compared to the X chromosome. And also the accumulation of those repetitive sequences. So that's what's similar to we observe today. And with this process, the number of genes also reduce on the Y chromosome, and we'll see some of the possible mechanisms of the Y degeneration in the later slides. So in this process, they try to illustrate our hypothesis on how the Y chromosome become degenerate, and also to explain the divergence between the X and Y chromosomes.